Okay, so hopefully you watched the animation and looked over the list of proteins and enzymes that function in semi-conservative replication. And then you can look back at this picture and make some sense out of it. The order in which the enzymes function, if you memorize the function of the enzymes, the name of the enzyme, the function of the enzyme, and the order in which they work, you basically have learned how replication works in detail. So in this picture you have helicase. Let me get my pen here. Helicase is shown as this little triangle unzipping the double-stranded DNA. And then you have the primase laying down the little RNA primers. And then you have the DNA polymerase III extending from that primer. DNA polymerase I going back and removing the RNA primer and putting in DNA instead. And then ligase joining the Okazaki fragments together. And that's the main part of the synthesis. You do have some other proteins. The topoisomerase works ahead of the helicase. It moves ahead of it to keep the parental DNA strand, the original DNA double helix, from getting knotted or supercoiled. And then when helicase separates the two strands apart, the single strand binding proteins help to keep them apart until the primer and the new strands can be built. So you have a couple of extra things, but fundamentally, if you learn the function of each enzyme and the order in which they function, you have an understanding of semi-conservative replication. And so you do need to know the steps, and I will have a little activity after this lecture segment to help you practice that. And this is the way your book defines each of the enzyme or protein and their function. But let's get on to some kind of interesting implications of DNA replication. Of course, replication precedes cell division, nuclear division. It happens in S phase during the cell cycle. In meiosis, it precedes meiosis 1. And it turns out that there can be mistakes made during replication. So if a DNA polymerase puts the wrong nucleotide into the new strand, that does happen occasionally. It's a pretty, it's a process with a lot of fidelity. That means it usually makes the right selections in nucleotides, but occasionally it puts the wrong one in. And so the DNA polymerase is, really we blame DNA polymerase for probably about 2/3 of mutations that happen. And then there's environmental agents, environmental things that can cause mutation. So UV being a main source of mutation, X-rays, certain chemicals. So there are certain mutagens that can also cause mistakes or changes or damage to DNA. And the good news is there are repair systems. The first repair system is the DNA polymerase itself. The DNA polymerase has what's called a proofreading repair activity. It can, if it makes the wrong insertion, so for example here, the polymerase is moving along starting from the 5' direction, moving towards the 3' direction, or adding new nucleotides to the 3' end, we say. Then when, let's say it added the A across from the T, and then when it looked at this A, it added a G. So that's a mistake. So if it senses the mistake, it can turn around and cut that one out. And that's called proofreading repair. And then it'll look again at the template, and it'll see an A, and very likely it'll add the correct nucleotide the second time. This is called proofreading repair, and we think this actually corrects almost all the errors that DNA polymerase makes. But the proofreading repair can fail. And so there's a second system that can come in. And this is called nucleotide excision repair. And NER will repair not only mistakes that are not corrected by proofreading repair, but also damage that's caused by external agents like UV radiation, chemicals, and such. And what it does is it comes in and it cuts out, you can see with the little red triangles, it cuts out not just the part that's damaged. In this case, there's a covalent joining of two thymines, which bends the DNA backbone. But it'll cut out even some of the nucleotides on either side of the damage or mutation. And so this is an overly simplistic picture. I think it cuts out like 25 or 30 nucleotides in total. And that just ensures that it will cut out the mutated or the damaged portion. And then DNA polymerase will come in and just fill in the gap, resynthesize it, and then ligase will seal the final phosphodiester bonds. There is a hereditary disorder, an inherited disorder, in which individuals cannot make the enzymes needed for nucleotide excision repair. So what happens is DNA damage from UV can never be repaired, and it leads to lots of cancer in the skin cancer at a very young age. This is called XERODERMA pigmentosum, and it typically, I think the data says that 10% of infants age 1 who have this disorder already have skin cancer, and I think it's by age 20, I think it's like 90% of individuals who have this have skin cancer. So luckily it's pretty rare, but it's interesting academically because it's an inherited disorder in which you cannot repair your DNA. And so the repair systems, the proofreading repair, the nucleotide excision repair, they work very well, but like anything, there is a failure rate, and occasionally there will be a mutation, a change in DNA nucleotides left in the chromosome. And so that's a mutation. And if the mutation occurs within a gene, then we would call that a new allele. Certainly some new alleles don't have any different effect than the original allele, but some of them can have can be different, can create different effects. And so you may recall that that allele may be a dominant or a recessive. That depends on how it interacts with other alleles. But mutations are just changes in the DNA.